we really want to take the last hour or so here to really focus on the science. So we wanted to get those individuals who are actively involved in this kind of research to join the panel now. So thanks to everybody who participated before the break, and I really do think we got some great perspectives to jumpstart the discussion. So we're going to pick up right where we left off. So, floor is yours.
I'm John Cross, and I'm an advisor to the Defense Department. And what impressed me the most from what I've heard earlier today was the increasing fidelity of these three-dimensional models, or four-dimensional, if you define it that way, to the human body. And I was particularly impressed by the talk from the lady from Switzerland, Dr. Boss, regarding the fact that you can now model a distinction between between cells treated and untreated with trovafloxacin. That's a very difficult to detect kind of toxicity. It was detected in the human population after the drug was on the market and had to be withdrawn. But my observation, my question has to do with the trade-off that several people alluded to between biomass and sensitivity for analytical instrumentation on the one hand, and the flow of the medium. I saw only one case where the rate of medium flow was compared to the normal human body, and was like a 1/10 of the normal human body through this microfluidic system. So I'm wondering whether I've seen a lot of evidence that cell cultures were, these 3D cultures were performing like the native body in a variety of different high-tech ways. But I haven't seen the old-fashioned measures of oxygen pressure and CO2 concentration and ATP. Is that because all of these measures are perfectly good and you don't see any problems? So that's my first question. My second question is, with all these microfluidic systems you're using now in many cases, presumably you could program the normal cycles of eating and fasting through the day. And I wonder if anyone's tried to do that.
Well, I'll take the first question. So the next phase of what we're doing is actually incorporating chemical analyzers onto the devices to look at zonation in the liver. So we'll be measuring oxygen gradients, glucose content, and that's adding some electronic sensors that John Wixwow at Vanderbilt, who's part of our team, has been working on. We just haven't implemented them onto the devices yet, but those are things that have to be measured. Because in fact,
What about ATP?
ATP has been lower on our list, but certainly the goal would be to measure as many parameters as possible.
Similarly, I think it's important to understand the metabolism that's going on. I think that's sort of the question with ATP generation. We have to understand better the metabolism, the cellular metabolism that's taking place under different conditions in these MPS systems. So the system that John Wixwow is developing, a microclinical analyzer that can analyze in real time with very small quantities, several nanoliters of fluid. It can continuously read out glucose and lactate and oxygen. So it doesn't read out ATP, but it gets you pretty close to looking at metabolic activity and metabolic function. From the kidney standpoint, it's very important, and we still don't really have an answer for this. To what extent are we getting oxidative phosphorylation versus glycolytic metabolism? You know, proximal tubule cells are very energy-consuming, energy-producing cells, chock full of mitochondria. We know that in 2D cultures, there's almost no oxidative phosphorylation. Historically, when people put proximal tubule cells into culture, it really doesn't replicate the metabolic activity of those cells in vivo. So I do think these are important parameters that we haven't fully gotten to at this point in time. In terms of your second question, I think that's kind of an easy answer. We have not, and I suspect that to date it hasn't been done yet to really vary nutrient delivery over time to try to generate something that resembles a human eating cycle as far as nutrient delivery in these different systems. The other issue you raised was flow and scaling. We are thinking a lot about that, and John Wixwell, I would say in particular, has thought a lot about how to scale the function of these different organs together, and whether it should be done based on their biomass or it should be done to their importance in terms of, let's say, drug metabolism, how best to do that. But we've replicated in our system, we have a flow rate of 1/2 to 1 microliter per minute, which scales to about, and that's per nephron. So we're basically recapitulating one nephron, and there are 2 million nephrons, 1 million per kidney in human kidneys. So our flow rates are pretty much scaled to renal blood flow rates, which are 500 cc's per minute across 2 million nephrons in healthy kidneys.
Okay,
Also,
I'm going to go with, yeah, go ahead.
I
I'm
just
sorry.
had a quick follow-up. So we're also working with John and also building inline sensors. And, you know, one of the other aspects is also inline mass spec. So we're, you know, with the flow rates and the volumes that we're getting out, we're kind of on the edge right now. But I think even that technology, you know, for prepping the samples is actually the biggest issue, desalting and pre-prepping it before you actually put them into the mass spec, because the mass specs can actually handle very small volumes. We're getting where we can do that, but we can also scale up. I mean, we do things on a microfluidic scale. We can actually do things on a little bit larger scale to make some of those assays easier to do. And one of the things with the oxygen, and I think you've run into this too, so, you know, PDMS, the typical material that we use for prototyping, is basically an oxygen sink. And so it not only is permeable to oxygen, it also actually absorbs it. And so you have to be, you know, we have to use different materials and ways to get around that, and thinking about some of these challenges with alternate materials to develop these kind of assays.
Okay, Joyce.
Joyce Suji, I'm a member of the committee. Christy Say's touched on this in her talk, and that is two systems that are very involved with toxic insults are the endocrine system and the immune system. What is being done to try to incorporate some sort of feedback from those systems with your models, I guess? For example, you can have cytokine release that results in recruitment of macrophages. Those macrophages then do things to the tissues, or then cause more cytokine release or more messenger signaling. And then the endocrine system is also complex. So you have not just you have signals going all different directions. You have flow going all different ways. I guess, what is being done in those areas, I guess?
So I can just say for our system, we try to make it very modular in the sense that exposure is always number one in our book for how we're trying to do the experimental design. And so for the pulmonary exposures, when we expose co-cultures of some ratios of the epithelial cells and macrophages, that exposure happens in a chamber, and then that comes out of the chamber and then goes into the microfluidic system, or even just a three-dimensional system without any fluids in it, where the dendritic cells exist at the bottom tray, and then we move forward with whatever post-exposure time period we want to use or hook it up to other systems. My biggest issue and problem with it that I scratch my head is just the clearance. And this is mostly because of the particles that I work with. I do this a lot with a lot of vapors as well. But with the particles, you know, that I don't know how to think about, or what engineering techniques the experts have for trying to get, if we can do anything about clearing the particles out of the system, and can the microfluidic system do that after the particles, for instance, are engulfed inside of cells? But we do a lot, we do exposure, and exposure is an experiment, and then the incubation post-exposure has different cells. We think of it as two different things. Everything's compartmentalized for us.
So I would say classically, in experiments, what scientists like to do is vary one parameter and see how that affects the output of the system. And I think the challenge that we've all had in this is there are many, many, many parameters. And I think I put one slide in my presentation that had about a dozen different parameters ranging from the cells to the perfusate to the seeding to the flow rates to the matrix. There are many, many, many different parameters. So it's really challenging to optimize these systems. And your question about what hormones or what growth factors or what cytokines are going to affect the output of these systems is a real challenge. And we've tried to, for each given cell type, in an empirical way, try to optimize what's in the media, what's in the perfusate. But by no means have we had the time or ability yet to systematically do what you'd like to do in science, which is have your whole system optimized except for one characteristic and then vary that characteristic, see what it does, and go stepwise through. If we did that, it would take an extraordinarily long time given the number of variables in setting up these systems. So we just sort of make our best judgment right now for what to put in the media perfusing these different organ systems.
So, we definitely want to add back the immune system components. And I think, I showed you the inflammatory response where we can add back the neutrophil. It'll be a little more complex when you start looking at more immune responses, because then we probably will need to have those components, the immune cells as well as the tissues, from the same individual. Otherwise, you may be looking at something like a rejection model, correct? But an interesting anecdote coming, starting off at simple. So in Dan Hu's Science Translational Medicine paper on the pulmonary edema model, you notice that there were no immune cells in that model. And kind of the dogma was that you needed the immune response was critical for that pulmonary edema to occur. And so the reviews came back, and one reviewer said, "This is horrible. This is an artifact because there's no immune cells." Another reviewer came back and said, "Hey, this is great. It shows that immune cells aren't obligate for the response." And so by starting simple, you actually learn something about it, that the immune cells weren't obligate, but I'm sure they have a definite effect, and now we can add those back and actually look at a secondary effect. With the transport of the nanoparticles, in the original science paper that Dan Hu published in 2010, so he looked at like 30 different nanoparticles across that alveolar endothelial cell interface. And one of the interesting things was that breathing motion was really activating a transcytosis process where you saw clearance from the upper alveolar compartment to the vascular endothelial compartment and outflow. You could actually measure the nanoparticles in the outflow. In a static system, there was very little transport. And with the active mechanical activation, it was almost tenfold increase in the transport across those. So one of the things I think is setting up the conditions correctly, but I think with the microfluidics, you can actually start to look at those kind of transport properties and clearance of the nanoparticles.
It's good to know.
And I just want to add that the female reproductive tract is in itself a great endocrine system, and not just the follicle itself. The biology of the uterus and the cervix is that it releases a ton of cytokines and growth factors already, and that was synergizing with the hormones that are there, and that's how the physiological effects occur. So I feel like starting at a reductionist level, making it as simple as possible, trying to understand it. They release their own stuff, and then we figure out what it does. I think we're at that stage right now. But in terms of the immune cells, I think it's going to be very important when we want to study things like HIV transmission that I was alluding to a while ago, where immune cells there in the endocervix is going to be very, very important in allowing transmission of the viruses, et cetera. So it depends on really what it is, the endpoint that we do want to study. And right now, we try to keep things as simple as possible and then build on top of that. It would be wonderful to get some blood vessels in that uterus, because the uterus is an organ that sloughs off every month. I mean, that's the biology of the uterus, and I feel like we're missing a really key component right now. And so as soon as we put in blood vessels there, it's going to change that cytokine and growth factor repertoire then it's going to cycle over the month, and so it's going to complicate things even more. But
Monita Sharma, PETA International Science Consortium. I have a two-part question. First is, how far are these systems from predicting chronic toxicity to chemicals? And the second question is, in a couple of the presentations in the morning, it was mentioned that you can culture the cells for long term in these kind of systems, 20 days, more than 20 days. But then given the chromosomal aberrations or other changes that the cells undergo, when you culture them long term, how efficient are the results in predicting the long term effects? Thank you.
Well, I guess I can start. In terms of, I mean, it's just going to depend on what cell type you're talking about, what toxicity you're talking about. So that's a hard one to answer. The models do show that they can be maintained for a long period of time, whether it's cardiomyocytes or hepatocytes or neurons. And almost in all those cases, they're post-mitotic. So chromosomal abnormalities are highly unlikely, if almost impossible in that environment, unless they're really going through rapid accumulation of ploidy for one reason or another, but typically they wouldn't be. So chromosomal instability really is a feature related to more of a rapidly replicating cell type, like a iPS cell, which in all these models, you're terminally, at least if they're using iPS cells or primary cells, there's no pluripotent cells in that mixture. So it's I'd say not a relevant concern. And in terms of predicting toxicity, that's generation 2 or next step type questions that have to be asked with all these models once they're up and established and show the sort of baseline activities and baseline properties are close enough to what they are intended to look like. Then the next step is to throw a host of chemicals at them and see how they respond.
Okay, Cheryl.
Walker. So I had a question about the fourth dimension, and unlike Christy, for me that means time. So a question to Kyle and a question to Julie. So when you make an iPS cell, does it think it's a young cell? Does it think it's an old cell? Does it think? And then Julie, you may have said it and I may have missed it. When you were making your follicles, are they going through the whole cycle? Does it depend on where the cells come from in the cycle when you assemble them? I mean, how is the cycling set? So maybe the first question to Kyle, iPS cells, are they old? Are they new?
I'll try not to be snarky. They are, when you reprogram them, they basically recapitulate the embryonic-like state. So they have, they self-renew, they can make all the different tissues of the body. So they, in essence, are starting over. So they're new. So we, like I mentioned, we made an iPS line from someone who was in their mid-90s. I made an iPS line that was able to proliferate and do everything else and differentiate in different tissues. And we've made iPS lines from cord blood, so, and everything in between.
So let's say you're making a kidney. Is it going to be like a nephroblast? Is it going to be like a mesenchymal rest? Is it going to be a kidney epithelial cell of an 80-year-old? I wasn't actually being funny. I kind of, because cells change over time, right? And so when you start them, where are they? And then do they continue to change as the normal cell would?
So what kidney or liver or heart or in all cases for at least iPS-derived tissues, they all leverage directed differentiation starting from and recapitulating what happens in embryological development. And so they're using growth factors to drive things towards the sort of early lineage steps and then down into the, into the more terminally differentiated cell types. So by and large, they recapitulate what's going on in a developing embryo. So they look and act similar to that age of development, independent of the iPS, the donor's starting material.
So I'm going to give it to Lauren in a minute, but another reason for asking that question is we know so many environmental exposures it makes a difference whether you're exposed early in life or later than life. While the tissue is still developing, and the uterus is an example, they'll respond one way to an environmental toxicant. When differentiation is completed, they'll respond another way. The breast has a very long window of development. And so I guess what I'm really asking is, do we actually have a window we can control that would let us recapitulate these early life exposures that we know do very different things than when an adult tissue is exposed to the same environmental toxicant?
Well, so what we do know is that they recapitulate sort of embryo fetal development, and then they look sort of like an immature phenotype. And depending on what you do, and that's really why in our lab, when I was at Roche, we spent a lot of time looking at small molecules and looking at sort of epigenetic modulators, panels of kinase inhibitors, oxygen tension, all sorts of different ways to artificially speed up aging process. And at the same time, we were beginning to look at microphysiological systems. So I do think, and if you look at sort of developmental, the differentiated, directed differentiation protocols, nearly all of them use a small molecule here and there, a PI3 kinase inhibitor, a ROCK inhibitor, et cetera, to either increase efficiency or push them down the differentiation cascade faster. So I do think it's a matter of time before we identify things, but similar to how I showed for arrhythmia, you can predict arrhythmia very well with an embryonic phenotype or a neonatal phenotype. If you're interested in sort of age-related heart failure, that remains to be seen whether that model will work for that until you get them aligned and you can then reproduce that aging aspect. We've done a lot of work on diabetes, fast onset diabetes, and showed that you can reproduce what looks like in an aged diabetic heart fairly quickly. So it's going to come back to fit for purpose and what's the goal, and does my model show that phenotype, and then how do you either speed it up or bend it to your needs. Basically.
So I'm going to invoke moderator's privilege for just a minute and ask a follow-up if I can to that, and it's really a technical detail question. Using these platforms, have individuals or groups taken, I'll use liver, but if there's another example, let me know, an iPS-derived differentiated cell and compared that in the microfluidic system to what was originally a primary human cell, and do you get the same thing? Do you get the same result?
So I don't have an experimental answer yet, but that's exactly what we're starting to do now.
So I think scientifically that would be something worth knowing, because then it helps to establish if there is a preference to which of the systems one would want to use.
And there's
and probably a lot more than that.
You can make it more complicated, and that is you can either add in the differentiated iPS cells into the device, or you can actually let them age in addition to the treatments within the devices. And early studies suggest that there's going to be differences between those two approaches. And it's going to be more, we think, physiologically relevant that they're aging within the device in the presence of other cell types.
Okay.
I had just another added wrinkle, and that's whether or not you've looked at twins, and what would you expect there in terms of IPC?
In terms of, well, I can tell you an anecdote. So as part of the CERM grant, they've been beginning to manufacture IPS lines. And these are all, the way that grant works is we just make the IPS cells, Coriel does the banking, and then 7 different academic investigators in California submit samples from diseases that they're interested in. And so one of the first runs they're going through, and they're doing the quality control, looking at sample identity and ensuring that there was integrity through the process. And they got to the end, and I think it was like 25 lines, and like, oh gosh, 2 of them are the same, based on this quality control metric that we're using. And so they start panicking, like, let's go back through the data control records, let's look and see how did it get switched, is it in the automation processes, et cetera. And then they call the investigator, and turns out there's identical twins that were in that study. So it was a good thing because the quality control process actually identified this, and it also is a good thing that there's identical twins in it. So an anecdote, hopefully that answers the question, but ideally if you're making a reprogrammed line and they are genetically identical, they should be syngeneic, similar.
What was the readout? Was it genetic readout or was it phenotypic
it's genetic. It was genetic. Had to be.
Yeah, so
I'm going to ask you guys to get up and go to the microphone because you're now consuming time. You can answer, but thank you for your patience.
so Cheryl's question, we do start with either primary or secondary follicles from the mouse. Does that answer your question?
Oh, so they're already set in the cycle.
Right, right, yeah, and then we mature them in vitro. Mm-hmm.
I wanted to make a brief comment about a previous question, a few questions back, about trying to deal with chronic situations. And so, I'm sorry, Rosemary Hunziker from NIH again. The NIH program is supporting a number of different systems, and some of them are similar tissues. So there are multiple liver, for example, platforms out there. And so different investigators like to get engaged about different questions. And so one of the other ones, which is not addressing some of the issues that Lance is talking about, comes from Linda Griffith's group at MIT. And something that she is keenly interested in and is doing a lot of work on is what happens when you challenge, for example, her liver tissue that has already been exposed to one toxicant or is from some kind of comorbid situation, and you bring in another second challenge. And so how does that change the response pattern of that particular tissue? Very complex question. I don't think that you would think, or she would think, that there are significant answers yet, but I think at this point what people are trying to do is decide whether their systems are robust enough to answer these questions and how they might change them and tweak them so that they can answer these questions. So I think that gets directly to the question that you had about chronic exposures and/or the kind of comorbidities that you would find across a population when you're looking at environmental challenges. So the NIH and DARPA programs are very, very broad in the conceptualization of the kind of topics that are being looked at.
Yes.
Maureen Bunger from Psychon Innovation. I'm curious about how you're going about choosing the media systems for these tissue chips and whether or not you believe the existence of a universal media that would eventually link them all together. Anyone dare?
So the goal, if you're going to start linking organ systems together, there has to be a universal medium. We have not focused on that ourselves at this time, but there are other people in the NCATS program, and I'm sure that you've dealt with it, that are working on that. In this next phase, we're going to find a, quote, universal medium just for the kidney, liver, and gut as a starting point, but that's hasn't even begun yet.
So it depends how you interlink the organ systems, right? So if you actually do it through vascular channels that connect, then you may be able to get away with a single media that's compatible with the vascular cells, endothelial cells that you have, or even potentially perfused with blood or plasma. Currently, we can't do that with all the organ systems, so you do have parenchymal cells being connected to parenchymal cells or the outflow of endothelial cells. And so kind of the simplest blood substitute that we've tried is basically a 50-50 mix. And for most things, that sort of works. So that's the initial kind of blood substitute. But there is within, just as Lance pointed out, within the DARPA program, an effort to develop a universal media or blood substitute. So, and that's going to be developed over stages. So it's not an easy issue, but, one that we're certainly considering.
Dave Jackson, DOD. I know mostly from long ago in the past about liver, so this is really what is driving me to ask the question. And that's how well the systems are all claimed to be differentiated. How well have you actually characterized the differentiated state in the cell lines that are growing in these systems? I mean, primary hepatocytes, they differentiate very rapidly. Often they appear to maintain significant aspects of the phenotype, enzyme activity, and so forth, but the transcriptional environment is quite different once the cells are placed in culture. And I'm just curious about where these things actually stand on the differentiation continuum with respect to a cell in an organism.
So this is something else that we've only just started doing because, and I give ourselves an excuse, because the best part of the NCATS program, it was milestone driven with timelines. And so we said we were going to do A, B, C, D, and E. And even if F was fantastic, and that's a good example, we had to put it off until we got into the UH3, this next phase. Now we have other things we're doing, and RNA-seq as a kind of a tool over time is a key element that we'll begin to implement.
Yvonne Rusin, University of North Carolina, a member of the committee. I heard Rosemary say something that made me get up and actually ask this question, because she pointed that, you know, in the large program, different groups are working on the same organ, and they tend to focus on different parts of the physiology. And that is great, but at the same time, it actually, I think, puts off the qualification of these model systems, because no one has a, fears that they are wrong, because they're just working on a different part of the elephant, right? So everyone is feeling the elephant from a different end. And also, as Rusty said, just reflecting on the, some of us who lived through the microarrays in toxicology and toxicogenomics excitement, I think we're very careful in perhaps, making promises about 1 to 3 year timelines, because I think we don't have Francisca here, and I think she was providing us with a little bit of a reality check, because if anything, a pharma company that is really eager to insource a lot of different technologies, they've done a lot of work and endured a lot of pain to actually bring in those technologies for the liver that are much farther along in terms of their commercialization. And I don't know how different people took her presentation, but I definitely was on the glass half empty side, because if you actually look through the tables that she was showing and some of the positive and negative controls from the in vivo toxicity, it wasn't very I think pretty. I mean, there are challenges, and you need to do, more detailed dose response and longer systems. But I just wanted to get your kind of, realistic opinion, perhaps maybe, not thinking about the milestones and then, trying to rush for the deliverable, but trying to be more realistic about when these systems actually are going to take hold in applied sense, because there have been a number of liver on the chip, Heprogen and Xiaxal and Regenemed, that have been around for, I think, at least 5 years as far as I can remember, and there's still, the reason I asked the question of Francisca, how much of their throughput they're replacing, the answer that I heard was very little, and for a particular application. So I would like to hear perhaps, maybe some of the glass half empty assessment and, not necessarily self-criticism, but it's also very important for us to bring out the challenges, for all the liaisons and all of those people on the web who are potential users, so they really understand, the reality of things.
I mean, my feeling is, if I compare the sort of experience base I have with cardiomyocytes and arrhythmia, where the electrical channels are all there, and there's no leap of faith to show that an MEA or some electrical event that occurs in vitro can have a very clear correlate to an in vivo sequela, to a drug-related event. So there, that part has a nice, easy story that's easy to trail, and especially if you're only worried about that electrical aspect. You're not worried about whether it secretes interesting factors, whether it has its force of contraction changes, or if it is undergoing heart failure. None of those questions are relevant. You're asking a question about arrhythmia prediction. And so there, that stepwise biomarker activity has a nice clear path. When you talk about something like liver, that can mean, is it an endocrine organ? Is it a target for hepatitis? Is it a factory for P450s? Is it a transport entity? Is it suffering because the Kupffer cell did something weird to it? I mean, there are so many different possible events, especially when you talk about from a toxicological validation perspective, that makes it difficult. So basically what you need to go through is how people are going through it now, is is it better than what we're using today? Is it going to help me answer a question that yesterday was either impractical or just wrong? And walk along that path slowly. I just think it's going to be very hard to say, in a year's time or even 2 years' time, to say this is a in vitro liver organoid, and it can do everything that an in vivo liver will do, better than what we, better than an animal would, for example. So, to me, that's the bar, and the bar has to be driven by stepwise experiments that ask very targeted questions about which particular biological property am I trying to reproduce, and how do I assess it?
So I'll answer further that the earlier question was about when things would be available for testing. So there's two answers. One, when do we think, I think, or any of us think, that the devices are robust enough that with protocols and QC characterization and cell lines and flow rates and all the, what I would call the basic engineering characteristics, are good enough that if I sent all of that to you or you or you, that you'd have a decent chance of reproducing some of the basic things. We would like to get it to the point where it is robust enough and reproducible enough that in fact it can get in the hands of lots of investigators to ask all these exciting biological questions, where we'll find out whether the simplest design, for example, of a liver with four cell types is complicated enough to do a large number or just a small number of liver kinds of efficacy and tox testing. So our goal is to make it robust enough so people can explore it. I don't think that's today. I think it's going to take, at least for us, another year to get it to the point where we would say, use these cells from Coriell or CDI, use this media, use this flow rate, and the device engineered in a particular way. In terms of when it's going to be useful, and Francisca and others in the industry would use it as a tool, I think that's the starting point then for them comparing it to the simpler 2D and 3D devices, and that's N amount of time.
Roger Curran, Institute for In Vitro Sciences. Most of you, when you're developing the platforms, as I understood it, started out with continuous cell lines, tumorigenic cell lines, transformed cell lines of some type, and then moved, as you would want to either primary tissue or starting to move to primary tissue or IPS cells. Would you suggest that that's a good way to go for others starting in the, let me start from a different area. Have you found that to be actually problematic? Have you had to tweak things so much going into the normal human cells that you might now say, we should have started that way in the first place and it would have moved things further? Or are you is that a reasonable way for someone else to start looking at the questions?
For our system, we've never used cell lines because there are none. And the normal system is what we use from surgical specimens, primary. And even investigators that do 2D work, it's usually primary cultures from tissues, only because the cell lines available are cancer lines or, there just are none. So to answer your question, cell lines are problematic, but there are uses for them. If we had them, we could have used them to work out certain parameters, but we don't. So I didn't answer your question, did I?
Well, you answered that it wasn't applicable to your situation.
Right, okay.
So, for the systems that we're developing, we've kind of taken the cell agnostic approach, and so we'll use whatever gives us the phenotype that we need. And hopefully, one of the take-home messages from the talk I gave this morning is that these microfluidic systems and organ chips can actually provide the proper microenvironment to change the phenotype more towards what you want. So even taking things like, I think Cheryl asked the question with the alveolar cells, we used initially cancer cell lines, A549s, H441s. And then once you put them in the proper microenvironment, they actually did things like secrete surfactants, behave more like the native phenotype than they do in 2D cell culture. And I think that's a common thread that we've seen, even with the gut chip, CACO2 cells. They don't do anything in 2D culture that we see in our microfluidic with the applied stretch. So, Don, and people up here probably heard Don say this a million times. His typical quote is, there's no bad cells like no bad kids, just bad environments. And so if you put the cells in the right microenvironment, they actually behave more like the normal phenotype. And hopefully if you put, primary cells where we can get them, or IPS cells, they'll behave more like the normal cells.
So if you take them back out, they're not tumorigenic then?
Well, yeah.
I'll just add a comment to that, and that is we had a mandate to use human cells, and so we had no choice. And I think that was actually a good idea because it pushed everybody to start with primary human cells wherever possible. One of the things that once these platforms are available that other people can try, one of the things that I would like to have been able to done, and I'm not sure we have the bandwidth to do it now, but somebody should, is to take rodent cells and make the same system based on rodent cells where then you can compare directly to the whole organism response. And right now you can't take a human system and kind of correlate it with a rat, but you could correlate a rat microphysiology system to a rat. And I think that'll be exciting in terms of the predictive power of the platform to the organism.
Yeah, we're actually starting to do that now. We're looking at putting rat, dog, and human on our chips, and then comparing those to the. We have a pharma company that's interested, and looking at species difference. Obviously, they have compounds that have different species responses. And actually, in Vaughn's question earlier, so we do have like agricultural or agrochemical companies coming, and some of them actually want us to develop an insect on the chip, and so you can look at insecticides, insecticide gut, against their gut versus human, and actually look at efficacy and safety in multiple chips.
Yes. Hi, I have a fairly broad question for all of you. I'm Barbara Cleaforth with EPA, and a lot of the questions I was thinking of were actually answered along the way, so you've covered quite a bit. Thank you. But it seems to me that as far as ground truthing the kind of work you're doing and correlating it back to past work and making it more useful for agencies like EPA, there must be an awful lot of information out there from your stakeholders and clients and customers on past products, both from pharma and chemical manufacturers, from their failures. We all know there are so many of those out there, and that a lot of it's proprietary. But other than the biggies, there must be. Are there any avenues to making use of that old data? Any ways of getting a hold of any of it?
Well, I think it's a goal. You could create a 2x2 grid, safe, effective, animal models, human studies, and getting hold of the compounds eventually to test that were, let's say, safe and effective in animal models and either unsafe or ineffective in humans and discarded would be very important for us to test. The one thing you can't do is if something was found to be unsafe or ineffective in animal models, nobody brought it to clinical trial. So you can't really know. There are going to be some false negatives that you really can't do anything about. But 3 of the 4 components to that grid, you could test compounds. So I think there's an interest on the part of NCATS and on many of the programs to be able to obtain some of the compounds from pharma that failed in clinical trials, either because of safety issues or efficacy issues.
How about chemical manufacturers?
We haven't got that far, but that would be very helpful.
Yeah. Thank you.
So earlier today, what seems like a very long time ago, someone asked the question about the fact that the brain wasn't included in some of the systems, or the nervous system wasn't included more correctly. What I want to ask in light of that kind of discussion is to this panel, are there any organs or an organ system that you believe won't lend itself to being recapitulated on a microfluidic chip?
Well, there's really quite, you're hearing from 4 panel members, there's quite a bit of diversity even in the funded program. So there is a brain on a chip being developed, John Wixwow's individual UH2, UH3, cartilage on a chip, almost every solid organ on a chip. We've heard about bone marrow today on a chip. So I can't think of anything conceptually that would say that you couldn't approach a given organ system.
Anybody else want to follow up? Everybody's agreeing? I told them they had to go to the microphone.
So I think that's a very interesting question. And, you know, I'm thinking about space travel and lack of gravity on bone being one of the huge major limitations. And I'm just wondering what we might be able to discover about if you can grow bone on a chip, the ways that we might be able to get around the effects of microgravity. Anyone thinking about that?
So that's a little more difficult question, because then you're looking at compressive forces. And so, you know, potentially there are ways that we might be able to engineer that, but I think that's, you know, farther out. But it's an interesting question. We thought about that, but it's just, you know, the experimental protocol to do that and actually, you know, ask that question in a relevant manner, I think is a little beyond us at this point, but it's certainly, you know, potentially doable in the future.
So since I walked all the way up here, so is NIEHS, John, or others thinking about microgravity as an environmental consideration and working with NASA on those environmental health effects associated with microgravity?
My quick answer would be that in the current budget environment, not necessarily looking to expand the environmental factors. I'd be happy to talk with NASA if they were interested in partnering.
Good answer. There is a broader NIH program on it, though. Yeah, I'll talk later.
I think I feel like we maybe are starting to wind down just a little bit, but I'd actually like to ask this additional question to the panel, and again, it relates to just critically looking at the science that's in front of us. And I would actually like to ask each of you to consider answering what you consider to be the next milestones in the progression of the science and the technology that you're developing. So, you know, are there specific challenges that you see as potentially being rate limiting that if we can overcome them scientifically, that's going to represent a significant advancement for the use of the tools?
I think
And now we'll sing the Jeopardy theme while they think about it.
Yeah, I finally won. I was losing every time. I think the question that Maureen raised about media is a pretty valid one, and that would help. I think not only in terms of getting multiple organs to flow together, but also to do multi-cell types within an organ, within a tissue, within a same cell type. So I think that, to me, will help. That'd be the next big milestone, I think.
Is this working?
Yep.
I did. Okay. Just a follow-up question on that, and it kind of, in terms of milestones and development, and it goes back to the question, a couple questions that were asked earlier, which has to do with the lifespan of these systems. What is the longest lifespan of the system? Do we believe that this lifespan of these systems is, in fact, analogous in terms of telomere shortening and all of the other metrics of the real aging process? And is there a milestone out there somewhere in terms of our understanding of chronic processes? I mean, right now it just sounds like we're talking about 30-day subacute kinds of 30-day of a human cell is not a chronic model. What would be the milestone towards getting towards a chronic model?
I mean, so we've cultured cardiomyocytes for a year and stopped culturing purely out of boredom. So you know, if the environment, and I think these microphysiological systems will show the same thing. If you get to the point where the cells are content and they're not compromised, you can maintain them indefinitely, or you know, within reason, but longer than you have patience for. Yeah.
Yeah, I mean, we've looked at least several months, and again, only terminated the experiment because we wanted to get an endpoint. I don't see any reason why we couldn't maintain these for up to 6 months to a year. But it's an interesting question, is looking at like telomerase length and things like that. Is how are they changing in culture is a very interesting issue, which is something that we should look at in the future.
My answer is much more simple-minded, but I think it's very important. And that is, as we vary things, whether it's the stem cells, what other cells are around the stem cells, we're using them, what's the flow rate, what's the dimensions in the device, especially if you're varying dimensions. The use of a microphysiology database that's shareable across the internet, where people can get used to logging in the details. What was the behavior of the cells before they were put in the device? The genetic profile, the functional profile, and what was it after it was in the device, and over time, managing what is a huge amount of data even for a single organ system, but certainly when you're doing multiple ones, and the effects and the variables, I think is enormous. And we won't get to the point of understanding if we have enough control or enough sophistication in the device until we have all of that data that we can interrogate.
Jonathan, Julie, Tony, challenges?
I think for us, it's going to be hands down getting some iPS cells to differentiate into the reproductive tract. I think the availability of human reproductive tissues is going to be a struggle for anybody that wants to continue doing this. Other than growing mouse follicles, I think as long as you have some mice, the follicles will be there. But in terms of studying some downstream tissues, we can't always rely on surgical specimens. Some of those, most of those have diseases, and that's why they're having surgeries. And so I think getting those cells from iPS cells are going to be critical, and also a huge challenge because it's never been done. So can we talk later?
I also think the cell source is probably the biggest obstacle. We really have two parallel directions, or not parallel, going almost orthogonal directions, and one are primary cells and one are some kind of progenitor cells, and different groups are using either one or both approaches. And they each confer different information, but at this stage of the game, they're not really running in parallel, and we really, as far as I can see with iPS cells, we're not at the point where we can say that for many of the readouts that we're going to want in these complex organs, that they have a mature phenotype. They tend to arrest development somewhere between a fetal phenotype and an adult phenotype, and I'm not an expert in the field. But it seems to me there's still a dichotomy, and for some of the organ systems, like you were talking about, Julie, primary cell source is a major challenge. So getting resolution on the best cells that will be available to scale these systems up in the long term, I think, is still probably the biggest scientific challenge that's out there. I think the media is probably the second biggest challenge, and that will be a particularly big challenge when it comes to integration of more than one organ system together.
And Tony, you have the last word.
So I totally agree with what everybody's said previously as far as the challenges, and I think one that was highlighted in the earlier talks too was the missing organ. So I think that's going to be important to integrate things that are, especially when we start to interlink these organs together. There may be things like the endocrine system that we need to at least plug in an artificial manner before we can actually get that system online. But I think one of the earlier talks this morning too is the modeling. I think there's going to be a huge amount of modeling effort in this type of program, especially as we integrate the systems together. One is to model how the system is behaving within the microenvironments. So as we integrate more organs and systems, the PK, the clearance, the PD relationships. And then secondarily, taking the data and the endpoints, the relevant ones, and actually translating that to clinical endpoints in man, I think is not going to be a trivial pursuit. So, how to make that in vitro to in vivo clinical translation, I think will actually require some modeling efforts to do that also.
If you want to end it, it's fine.
No.
No. Okay.
We're scheduled to go to 5, so I'm not going to, I don't necessarily think we have to stop at 5 o'clock, but
It can
if you
be
have a
quick.
burning question, you have the microphone.
It is burning, and it can be quick. Christy Says from RTI. John's comment made me think of this. I came into this thinking that a lot of the systems that you experts were going to describe were actually going to incorporate, and maybe you are, and I'm just not putting it together, but harvested tissues into your system. But we keep on going back to iPS cells, or even Julie, you mentioned that you have the tissues, you digest them, and then seed them as cells, as opposed to tissues from cadaver or whatever, then plop into the chamber that you're working with. So I wonder if you could speak a little bit toward the construct and moving, to tissues, or maybe that is what you're doing and I'm not understanding some of the technical things you're saying. Thank you.
So yes, that is how we're constructing our tissue from cells, and that's only because we don't have enough when we actually get the specimen. If we had enough, we would seed them directly, etc. We have tried tissue pieces. It works well for the tube, for the fallopian tube, but it does not work very well for the endometrium. They don't do well in culture. They tend to get necrotic, and I think it is because of the biology of the tissue. It's supposed to be short-lived anyways, and so that's why we decided to create a more robust system and try to build it within a scaffold, and so that it will last longer. And remember that tissue also has everything else in there: immune cells, blood cells. So we decided to just take the essential components that respond to the hormones first, and try to build that up, and then go from there.
So does that imply go from there? Does that imply you may, all of you, are move to the tissue that would include the more immune cells and all the other components of the tissue? Or again, you're going to be limited by just the amount of tissue you can get from that particular organ site?
Right, so there is a limitation, and we are going to continue doing tissue work, and I think getting that baseline data on how they respond to hormones, et cetera, is going to be very important. But in moving forward, we do want to be able to have an endless supply of cells or tissue that we can make tissues out of, that is more consistent, that can be frozen down, available whenever you need. But I think having the tissue data is going to be very, very important to have that initial characterization of how the tissue should be responding, and we can definitely compare.
And the complexity of what you start with, a number of cell types, again, is what question you are answering, right? So it's fit for purpose. So you start off simply, and I think you build in the complexity as you go along. Otherwise, you have a system that's overly complex, and you may not be able to make any heads or tails out of it anyway. But I think Kyle pointed this out in his presentation. It depends on the tissue that you're trying to mimic. So heart, it's going to be very difficult to get primary tissue and actually grow that in vitro. It's just not going to be feasible. So I think the IPS is probably the best technology that we're going to be able to go down that route. There are other tissues, intestinal tissue. We've started to look at getting biopsies from area hospitals, developing organoids, and then putting those in our microfluidic chips. And we're having some success in developing monolayers out of those. So I think where you can make that direct comparison, and it goes back to Lois's question, we're going to try and do that. But I think it's going to be difficult with some tissues, and primarily because we want to do it in human. We don't want to do this in animal tissues.
Yeah, I guess to go back to a little bit what we touched on in my talk is hopefully all these models will eventually get to the point where they can be used to make individual decisions about individual either treatment paradigms or understand responses in individuals. And maybe at the end of the day, iPS cells or genetic diversity won't be meaningful enough to merit that. But my feeling is that for a vast majority of things it will. And you'll want to have a genetic background of interest as you're exploring that disease process or activity. And so to do that, iPS cells is by far, there may be a new technology out there that will replicate it, but it's by far the most amenable to addressing that question, on top of the points I made about genetic engineering. So if you want to do things like Lance does of introducing biosentinels, if you want to be able to tag the cells, identify them, unless it's a replicating primary cell that, that maintains its phenotype, which is relatively few, you're going to need to have some type of model that has a pluripotency stage to it that then can be differentiated into a tissue, then can be add back the additional components. So I look at it as, you know, maybe working out some of the conditions and variables in primary cell culture, but ultimately the question, if it's going to be valuable, we'll need to leverage iPS cells or something similar to that technology. And that's really why, and granted, it's a big conflict of interest, but that's why I believe in that technology. And that's why I think a number of the projects that were spoken about today are looking at that at some stage down the road as the final piece, is to have iPS cell-derived tissues driving the answers that are going to be gleaned from that model.
Rusty?
So in addition to systematic exploration of some of these media components and trying to get a universal media, is there a parallel effort going on in exploring ECM type composition across different organs as well as within an organ? Because that's going to be probably as important as systematic exploration of the media components, because a lot of that's obviously, given some of the work we've seen with the kidney, as well as not necessarily what was talked about here, but also, you know, in the liver, driving zonal heterogeneity as well as other components of differentiation going to be just as important, and sort of driving that terminal differentiation across different cell types and different structures within the organ, I guess.
Well, I think if you look across the different funded projects, there at this point in time, there's tremendous diversity in approaches, whether it's an approach to extracellular matrix or what Christy was referring to, the cell types that people are using. And I think at this stage of the game, it's very healthy. And getting together every 6 months and sort of sharing progress and challenges as a group, Lance referred to this earlier, I think has been driving all of the projects and programs forward. With respect to, I wanted to come back to your question, Christy. I almost see two different philosophies within the consortium. There are those who are really saying we are top down, we know what we want the architecture excuse me, to look like. So Lance gave a good description in the MGH system. You put a layer of cells in one compartment, you put a membrane there, you put another layer of isolated purified cells in another one, and you develop the system that you think should develop to replicate organ function. There are other groups that kind of treat this more as bioreactors and put a bunch of cell types together and think if we put them in the right microenvironment under the right flow conditions, they're smarter than we are, and they will self-assemble in a way that, thank you, that will better replicate function than we can top down. And both, there's merit and value to both, and there are a lot of different experimental approaches that are being taken with the different organ systems. For us, just to use as an example, we can't take all the primary cells, mix them together, because the renal tubule epithelial cells will out-proliferate all the vascular cells, and you won't even be able to find the vascular cells. So we micro-dissect out the kidneys, pull out and put the tubular cells into a tubular growth media, the endothelial cells in an endothelial growth media, and then try to put them into respective chambers in the devices. So I think it's all, at this stage, still working out the right conditions. I really think it's important for people not to leave thinking that we've all optimized the various approaches to all these organ systems. There are a million open questions, including how you put the cells together, what kind of matrix is preferable for what function you're looking for, and I think we're still all, to a certain extent, in the experimental phase.
So I think on that note, we're in the experimental phase. It's time to wrap up. So I've actually taken a lot of notes here this afternoon. I first want to thank both sets of panelists for leading us through what I think has been a very enlightening discussion. So I want to thank you all, and thank you. and thank yourselves for, I think, for what has been, I think, a really great discussion. I'll just a couple of things that kind of caught my attention. We're trying to replicate a very complex system, but we have to start simple. And I think, you know, when we critically look at these systems, as we've done today, you kind of lose sight of how much progress we've actually made. And I think what I heard today was a lot of just fundamentally strong science to say, you know, we'd love to change 15 parameters at a time, but we all recognize that that's not the appropriate experimental method. And the complexity here makes it even more challenging for us. Interestingly enough, I think, you know, we are probably approaching some level of systematic execution of these studies, whereby there could be some common protocols that are being developed and potentially used. And this technology, I find it interesting, is actually putting some pressure on our analytical colleagues to help build systems that actually allow us to become sensitive enough to analyze what these systems are doing. But within the context of that discussion, I heard things like a microclinical analyzer, which could be useful in lots of ways, not just with these systems. And again, increasing the sensitivity of things like LC mass spec that we could potentially use in flow-through systems here. So I think that, you know, there's a parallel side effort in looking at the analytical methods that are necessary in order to pull these kinds of systems off. I'm going to quote Lance Taylor for what I think is an appropriate sense of the state of the science. And he said, "We are advanced, but we're still in the prototype. And we are high content, high value, not high throughput just yet." So I actually think that kind of sums it up pretty accurately. And I'll finish with, I think, just to reiterate a couple of things. When I ask about the challenges, we heard things like the consistency of media that's used across organs and across platforms. Clearly, I think one of the things that is evident from today's discussion is that the source of tissues that are used in these systems is critical, can be rate limiting, and the availability of pluripotent stem cells is certainly a must for this work, as well as really recognizing and understanding how different cell sources could influence the results we get. There is the case of the missing organ. That just sounds like a mystery to me, but nonetheless, I think, no, it speaks to the complexity of what we're trying to do and how we would mimic that, the need for modeling within these systems. And I think the other case was made for a very clear need to have databases of results that can be shared and evaluated. And that's something that's really going to help advance the use of these tools in a consistent way. So that's more or less in the nutshell what I've summed up from the last 3.5 to 4 hours of discussion. But that's not a trivial place to be, I think, in any way, shape, or form. So I think that concludes today's discussion, unless the administrators here have any last-minute information for the group for tomorrow. But again, I want to thank you all for what I think has been a great discussion.